This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1113, recorded on May 10, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Today we have a little weird situation here on TWiV. We had some technical difficulties recording, and so I am re-recording this intro at home, so it's different. And in a few moments, you're going to see me pop into the incubator, much like H5N1 virus popped into cows, I guess, because that's going to be our subject today. Joining me today from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. It's great today to be inside podcasting because while it's 53 degrees, it is pouring rain and will continue to do so for most of the rest of the day, I think.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here, and it's actually not bad here. It's 61 Fahrenheit, which, as we know, is 16 Celsius. Just light wind, but it's a wind out of the east, so it's probably weather's going to deteriorate here soon.
We have a guest for you today. He is from St. Jude Children's Research Hospital in Memphis, Tennessee, Richard Webby. Welcome to TWiV, Richard.
Well, thanks. It's great to be here. And we're, it looks somewhere 70, 80 degrees here with a bit of cloud and wind, but no rain. Yeah, it's that time of year here in the Mid-South where, it can be 80 degrees 1 minute and then thundering the next and back to 90 the minute after.
Good job getting accustomed to the Fahrenheit scale.
Thank you. It took me 20 years, but...
If you enjoy what we do on these programs, giving you real science, we'd love to have your financial support to keep them going. Please go to microbe.tv/contribute. Now, Richard, we'd like to talk with you today about influenza H5N1 virus and how it got into cows in the U.S. But before we do that, tell us a little bit about your education and training and how you came to the U.S. and eventually ended up at St. Jude.
Sure. I'm actually from a little bit further south than Memphis. I live in North Mississippi. So going a bit further back than that. So I'm a New Zealander. So I went to the University of Otago, which is sort of in the bottom half of the bottom island of New Zealand, which it never gets anywhere near 80 degrees down there. I went to university and sort of with an interest in biology, not knowing much more than that, but then sort of got sat down by actually an Irish fella who was in the Department of Microbiology. I think at the time we sort of, as students, you lined up at these tables and spoke to the advisors, and there was no line to the microbiology table, and he was a bit bored, I think. But he sort of inspired me with in his own sort of Irish way, and so he ended up in the microbiology department there. Yeah, and so as is typical in New Zealand, you start the same institution for your PhD. So I stayed in Dunedin at University of Otago and did my PhD in viruses, but actually iridoviruses, so sort of large double-stranded DNA viruses. The ones we were looking at were of insects. And so at the sort of end of that, wanted to head towards a postdoc and move towards something a little bit more medically relevant, and sort of, as is typical then, applied for everything in the back of Science and Nature, and then sort of ended up in Memphis, Tennessee, working sort of with my mentor Rob Webster, who is actually another New Zealander, but at the time I had no idea when I applied. He claims it didn't help my CV, but I'd like to think it did a little bit.
It probably helped a little bit.
Yeah. And so, and then came to Memphis as a postdoc, and here I am 25 years and 3 days later.
And as I mentioned in the pre-show, I've known you for many years, your work, but we've never met. And I always thought, Oh, why would they hire a guy named Webby with Robert Webster already there? It's so strange, right? And I always
Yeah,
thought, are they related?
I've had lots of that. So
And both from New Zealand.
Both from New
that's right.
Zealand, right?
That's remarkable.
My worst moment was actually on a radio interview. Was for a fundraising, St. Jude fundraiser, and I think they must have had the Rob Webster cheat sheet. So they said, well, so you've been at St. Jude for 45 years. I said, oh, and I went along with it for a little bit before I had to abandon.
All right.
So when did you start working on influenza? When you came to St. Jude?
Yep. So that was, yes, I got here in 1999, and actually, so at that time, my first project was on just I guess the year previously, that the sort of the triple reassortant swine viruses in the US had just sort of started taking off. So that would sort of change the face of swine flu in the US. And so that was my first project. And so
And a
What
lot
year
of
was
people,
that 2008 or so, or earlier?
well, 1998 was when they were first detected.
Okay.
right. Now, a lot of people think St. Jude's cancer, but it's also a major research center for flu, right?
Yeah, so it's I think at least the patient population here is pediatric cancer,
Right.
oncology, and other hematologic disorders. But the research enterprise here is a lot wider, and sort of Rob Webster was one of the early faculty members here and sort of planted the flu flag. And so there's a few of us here now: myself, Stacey Schultz-Cherry, Charlie Russell, Paul Thomas, etc.
So let's talk about influenza H5N1, and maybe you could give us a little history of when it came onto our radar and what it's been doing recently.
So yeah, I guess the first time we saw H5N1 was actually not long before I came to St. Jude. So in '97, that's when 18 people in Hong Kong were confirmed infected with an H5N1 virus. So that was really the first direct evidence that these avian flu viruses could go straight to humans. I think, as always, if you read a textbook, of course, it always shows arrows to a intermediate host before coming to humans. But that event essentially showed that wasn't the case. They depopulated all the poultry in Hong Kong in '97, so that got rid of that particular form of the virus, but it was still smouldering in other parts of the region. It went underground for a few years. 2004 was the next time it sort of popped its head in quite a different form. So the hemagglutinin is about the only thing that, if you follow the lineage of the virus we have now back to 1997, it's really only the HA that's similar. And then, so the next, I guess, major event in about '05, the virus was detected in a large die-off of migratory birds in the middle of a big lake in China, Qinghai Lake. Up until that point, it had been a Southeast Asia problem, but that sort of signaled the westward march of the virus into sort of other parts of Asia, into Europe and Africa, where it did its thing for, again, a few more years until somewhere, probably mid-2021, where, as flu viruses do, they get together with other flu viruses and reassort and make new viruses. And this, what we call the 2344B H5N1 version emerged. And for reasons we need to understand, but we don't, that particular form of the virus was really well adapted for migratory birds. And so the amount of virus in that population skyrocketed. It changed the dynamics in Europe from being a more seasonal, as the birds came through, disease to a endemic year-round disease. And I think that also pushed the virus over into the Americas, where it came here in the end of 2021. Again, it did what flu viruses do, got together other flu viruses and made new versions of that, and they spread through North America down to South America, and I guess it's a version of that we have infecting cows now. So it's been a long road. It's been 25 odd years of this virus to get where we are.
So do we have any idea, going all the way back to the first cases in Hong Kong, and this was something that had been in Southeast Asia, as you said, and obviously this is a virus that's been in birds for we don't know how long, but was presumably kind of in a small area and then started making this move? Is there I don't know if there's any sequence data or anything that people have been able to look back at what initiated that whole process before it started moving west.
Yeah, so again, it comes down to this process of reassortment. So we don't know the exact, I can't tell you it's residue, 345 in protein A, but, what quite often occurs prior to these sort of changes in the epidemiology ecology is this a reassortment event. So where the H5 picks up different gene segments from different viruses. So there was a particular combination of genes that really drove that expansion around that time. It seemed to be more within the domestic poultry, so it was a selective advantage in the domestic poultry. So again, increases prevalence and that population spread. And so again, that's why I think when we see H5 sort of sampling different gene sets and combinations, we get a little bit worried because it usually signals, maybe I shouldn't say that, it often signals something's about to change.
So you mentioned cows, and I remember a little more than a year ago, some stories about things like sea lions and other types of animals getting sick with avian influenza viruses. And can you talk about some of the infections of some of these other organisms? I don't think before that I knew that some of those other organisms could be infected. Before recently, I didn't know cows could be infected. And so what's going on with infections of some of these other organisms we don't think of with flu?
Yeah, well, I didn't know they could be infected either. So, don't feel too bad. I think what we're seeing is this increase in the amount of some of these mammal species being infected with this virus. The question is, have they always been able to be infected, but they just haven't had the opportunity, or is there something special about this virus? Again, it's probably a little bit of both. I think my gut says it's probably there's just so much more of this virus around. Again, it became so more adapted to wild birds, much more of it around. Many, many more sort of birds became infected. Many more of them got sick, sort of dropped out of the sky, and along came a sort of a fox or a bear or a mountain lion and sort of probably ate or at least chomped on that bird and got a huge dose of virus, and so became infected. The sea mammals, that's a little bit more of a head scratcher. Clearly they do share some environments with lots and lots of birds, but it does seem that at least in the sea mammal point of view, probably more evidence for some mammal-to-mammal spread in that host. Then we're to cows now, which now Ian Brown, who used to be at APHA and now at Pirbright in the UK, had done some work many, many years ago suggesting cows might be hosts of flu, and I used to make fun of him for that. So now I've had to eat my words, but so this particular virus is teaching us new things about flu that we didn't know.
So you mentioned that the epidemiology changed. It was seasonal and then it became endemic. And do we understand why that happened? Can we identify changes that go with that?
So some of the changes prior to that point, it was an H5, but it was actually paired, particularly in Europe, with an N8 neuraminidase, so a different subtype of the neuraminidase. And flu has a bunch of different H types and N types, and the dominant form at that time was the H5N8. It did switch out, so went back to the N1 form, and so it was a combination of genes, and clearly there's something to do with that combination of that particular version of the H5 with this particular version of the N1 that sort of worked magic from a virologic perspective. But it's an area where, of course, lots of people are interested in trying to understand exactly what it was about this combination of genes that really led to a supervirus.
Was it a switch back to the same N1 or to a different N1?
Yeah, no, good question. It was actually a different one. So it was different to the ones before. And it's... so H5 is often paired with N1, but particularly when it's in chickens, quite often the neuraminidase protein, the N1, when it gets into chickens, it actually chops a little bit out of it. So it's a shortened version of the N1. This particular N1, seems to be a bit more of sort of a wild bird N1, if you like. So it's a full length, doesn't have those sort of chicken adaptations. So, again, maybe suggestive of that's why it's done so well in wild birds.
So
So in
then,
wild birds, does it cause disease, or is it's not highly pathogenic?
Yeah, so it depends what wild bird. It depends if that wild bird, how many flu viruses it's seen before. So
Mm.
sort of the sort of typical hosts of, like the mallard ducks, the pintail ducks have seen lots of different flu viruses, particularly if they're, older ducks. They probably survived a lot of it, although there was, huge die-offs of even some of those birds when this virus first came through. But some of I think, the biggest impact has been on some of these other birds that typically probably don't get a lot of flu. I'm thinking things like vultures and even bald eagles, right? That probably don't see a lot of flu, so they don't have a lot of immunity. So when this virus did get into those populations, it really decimated some of them.
So why is there a difference in how much flu these different species have seen?
Well, I think it probably comes down to, what is the natural host of influenza? It is these ducks, really. So the migratory ducks. So why don't they typically spill over into these other species? It's a question I don't know. I think if you again look at some of the early work by a lot of people, and particularly in the US, so some of the Rob Websters, the Dick Slemons, the Barney Easterdays, when they looked at where influenza was in these populations, the highest prevalence was when they were up sort of marshalling to fly south. There was lots of young birds, so in the regions of Canada, where they just had their young, and so sort of lots of new hosts. Yeah, so sort of an outbreak situation. But as sort of those populations came further south, sort of for the winter, the prevalence of flu dropped a little bit. So maybe there's not quite as much exposure between some of these other populations and the virus, but now that there's so much more virus there, maybe it's maintained for longer as it comes down those flyways, and so there's more exposure. But that's a lot of hand waving on my behalf, and not
so
everybody.
now the
Go ahead.
the virus that's gotten into the cattle, is it more like the chicken or more like the migratory duck form of the virus?
It's more like the migratory duck form. So
Okay.
it's
So this is not from old MacDonald on his farm and there were chickens and cattle. It's more likely they flew past.
I think so. Exactly. I mean, it's a big question right now. How exactly did this virus find its way from a bird flying past into the cow and the udder of a cow? Probably. That's a big unknown too. But no, almost certainly, I think this is sort of a wild bird to cow transmission with probably maybe something in between.
So I don't know how much sampling is done in the U.S., but is it fair to say that many wild birds are likely to be infected? Do we have an idea of what fraction might be?
Yeah, I think it's changed a little bit. Some of the work early on during the early years suggested that some of these populations had ridiculously high, in excess of 50% of birds had this virus at one stage. So I think the level of population immunity because of that is quite high now. So I don't think it would be that high now in these migratory populations, at least.
I mean, many people ask us if it's okay to have a bird feeder in their backyard. Are they putting themselves at risk? What do you think?
I think it's probably okay. I think it's probably so long as you don't go chewing on those birds. I think it's okay. I mean, there's no zero risk to anything, right? So there is a
even podcasting.
Yeah. That's right. But I think the risk of that is incredibly low.
But if you find a dead bird on your land, you probably shouldn't touch it, right?
That's right, and I think that's quite often gets asked that question. What can what can people do? Sort of wild birds. Well, honestly, not a whole lot, but just consider that any sick or dead bird, particularly if it's acting rather weirdly, because these viruses do get sort of very neurotrophic, then probably a good idea to steer clear of it.
Are there cows in other countries that have been infected? Do we know?
No, it seems to be the US is the only one that has it. There's some evidence that maybe in other events that maybe flu has infected cows, but not this H5. No one sort of detected that. So I do think it's a really rare event. The virus.
When do we think this happened?
So the smart folks with have looked at the sequence that have been put out by the FDA suggest that this crossover might have occurred late 2023, early 2024. So it does suggest it was circulating for a period of time before it was actually detected. But maybe that makes sense, because if you get a cow with mastitis, why on earth would you look for flu?
Right.
And I guess what fraction, you may not know this, but is this something that's impacting a lot of cows or a very small number of cows in the U.S.? How
Right.
how big of a thing is this in all U.S. cows?
I would suggest no one actually knows the answer to that one. The official reports, we all can head to the USDA website and look. I think everyone believes that those numbers are only a percentage of what the actual number of infected herds are for a number of various reasons. But certainly it's probably widespread in some of those states that have confirmed infections, and probably in states that haven't.
Have there been cow-to-cow spread?
Yeah, almost certainly. I think this is being, at least on the farms, is being maintained from cow to cow spread. I would suggest that it's probably not the way people might think. We think of flu being a respiratory virus that spreads that route. I think there's enough evidence to suggest that this virus is not doing it that way. So, USDA has come out with sort of the hypothesis of some sort of mechanical spread, and I believe that as well. So there's something that's happening on these milking farm environments that's actually transferring it from one cow to the next. At least predominantly, yeah.
Maybe on milking equipment or feed troughs or something.
Yeah, so I think the milking is sort of one of the favorite hypotheses at the moment. Again, because if you look at a cow, the milk, the virus in the milk of an affected cow is very, very high titers and not much anywhere else. And so
no fecal virus.
No. I mean, you can detect some virus in other swabs, but it's nothing compared to the milk. And it's only it's not. Of course, nothing's ever black or white, right? So, but the majority of cows that are getting it are the lactating cows. So again, suggesting it's something to do with that milking parlor environment.
So it's utterly localized.
It's utterly localized. That's correct.
You mentioned mastitis. So do the cows have any other kind of disease, or is this really seem to be localized in that way to the mammary gland of the cow? Because that's weird too.
Yeah, that is weird, and I think that's the primary symptom, but it's not the only. Again, that's coming from me sort of sitting in my comfy chair here in Memphis, and a lot of this is secondhand information, but people have seen the cows. Some of them, and it does vary. So I don't, it's not a particularly lethal infection in these animals, but there has been some mortality in it on a couple of the farms. But there's a really big drop in feed intake, milk production, and so
in their herds in different states that are positive, presumably those are independent spillovers from birds, right?
No, it's actually not. I don't think it's everything. Again, by the smart people who are looking at the sequences, it really does look like it's a single crossover event. So there's been one crossover into cows, and then the rest of it's been sort of mediated by movement of cows, which again, I've learned a lot about cows in the past few weeks. There's a lot of cows that move about the country.
After this, I'm going to be Googling whether or not cows get respiratory symptoms and whether cows can get the sniffles.
Yeah, well, there's again some reports of some maybe respiratory, but you got to think it's possible there are other pathogens. There are other pathogens, of course, of cows that do have respiratory origin. So,
but this was first picked up as a decrease in milk production, right?
Yeah, that's right. So from again, these guys are pretty smart. So they, a lot of these farms know exactly how much feed a cow is bringing in, exactly how much milk a cow is making, and so they can actually pick up those drop-offs pretty early on. And yeah, exactly right. That's what was the early indication something was going wrong.
Is the virus in milk infectious?
If it's coming out of a cow, yeah, absolutely. And it's, the infectious titers, we've done some of that. There can be eight or more logs of virus per mil in milk coming out of an infected cow. So yeah, the virus loves it inside that udder.
So maybe this is a new vaccine production platform we hadn't thought about.
It could be. Yeah, it's. I mean, it's. You have to crack open chicken eggs. Yeah.
Right.
Yeah, so pasteurization is taking care of it, though. Is that correct?
That's the current thought, right? So everything downstream of pasteurization that's been tested to date, at least that I've seen, has, there is no virus there. I'm sure many people have seen the stories of the supermarket milk that are at least positive by PCR for the RNA of the virus. We've tested a number of those as well, and nothing so far post pasteurization. But a lot of studies going on in that area too.
Now, at least how many people have been infected from cows? Do we know?
Just the one. Well,
just the one in Texas.
Do we know? Yeah. So there's one reported. Yeah. But again, it's the how many cows are infected, probably more than's reported. How many humans have been reported infected, probably more have been reported as well. But just the one was a farm worker in Texas who had conjunctivitis. That's a CDC paper just come out of New England Journal of Medicine, and this guy had some pretty bad looking eyes.
But as with the mastitis in the cattle, if a doctor sees that, they're not going to think flu as the first thing to look for.
Typically not, but we do know that flu has, particularly some of these avian viruses, have the capacity to cause conjunctivitis. We saw it, the royal we, in the early 2000s in the Netherlands, where there was an outbreak in chickens, and a lot of people in contact with those actually had conjunctivitis. There's even an old paper of a infected seal sneezing on a worker, and again, conjunctivitis was the main outcome there. So probably, I think your average GP wouldn't associate conjunctivitis with flu, but it's definitely known that this can happen.
So the what was I going to say now? Oh, besides cows, what other can pigs be infected? Have they been infected?
So on at least the infected cow farm, so if we think just narrowly talk about this outbreak, there have been some cats and raccoons who have been infected, presumably through consumption of contaminated milk. And of course some spread of the virus from cows to chicken farms as well, probably by shared people or so moved by people between the cow farm to the chicken farm. So far, pigs, luckily, no. And so, there have been some experimental infections of swine with these H5, the 2344B H5 viruses, which are sort of not the exact virus that's in cows, but close cousins. The virus, it can replicate, but doesn't transmit very robustly. So, luckily, I think right now swine don't seem to be a good host for this virus, similar to humans.
Could a different route of infection affect the severity of the disease? I mean, you've got a couple of farm workers with conjunctivitis. That's obviously not the way this virus normally spreads among humans, but I guess the fear would be somebody then spreads it via the respiratory route. Maybe they develop conjunctivitis, get viremic, spread it respiratory to somebody else. Is that likely to affect the severity of the illness?
Yeah, I would think so. If I had a choice of having this virus in my eye or in my respiratory tract, I know where I'd pick. It wouldn't be the respiratory tract. So, but I think you're dead right. This was it wasn't a mild case of conjunctivitis. Again, I'd tell people to head to the paper to look at the poor guy's eyes. But no, if this gets into the respiratory tract, particularly into the lungs of someone, then that's a completely different disease for sure.
So what sialic acids are these viruses binding to? Are they still alpha-2,3?
Yeah, so they still bind the preferred avian virus receptor 2,3, and there's actually a lot of that receptor in the conjunctiva of the eye, which is why it makes sense this virus is replicating there.
And I guess in mammary glands of cows also, right?
That's right. Yeah, so there's one fairly high level sort of look at that in terms of what sialic acids are in the udder of cows, and by sort of a group in Denmark, and they have found both 2,3 and 2,6 in the udder of cows.
So that's a little concerning, because if you have both there, then you have viruses binding to alpha-2,3, and maybe a variant arises, and then it gets amplified on alpha-2,6, right? That's a concern.
Absolutely, yeah, that is a concern, and we got to, at least keep in mind with this particular study, it was a fairly preliminary first look, and the 30,000-foot level, the true sort of glycobiologists, will keep reminding us that there's a lot more subtlety to that interaction of sialic acid and flu than just 2326, but it does sort of, to me at least, raise sort of the risk level that there is that possibility of changing the preference or receptor by replicating in the udder. Although again, the virus is growing to 10 to the 8 logs of virus in the udder with a 2,
how much better are you going to do?
Yeah. That's exactly right. Maybe it doesn't. If it changes, it would be worse off. So maybe there's this pressure against it. Maybe.
But that's a change. That's a change you would look for in trying to predict whether it's going to make the jump into humans successfully, right?
Yeah, absolutely. So that sort of receptor preference change from the 2.3 to the 2.6 is something we all believe that these viruses would have to make to change from being a duck virus to being, or from a cow virus to being a human virus. And we know pretty well where to look on the virus for those changes. So yeah, lots of people looking at that.
There's also a polymerase change that would be needed, right? Is that happening in these cow isolates?
So so that there is good evidence from both experimental studies and also natural infections of mammals where we do get changes in the PP2 polymerase component that's associated with replicated mammal cells. The human case with this bovine actually had that classic change. So the PP2 627E to K change, it did have that. The swine viruses don't have it. They do have another mutation that there's at least one publication suggested enhances replication in mammalian cells. I can't remember the exact name off the top of my head here, but it's all of the cow viruses do have that, but it's a little less clear in terms of whether that has exactly the same impact as a 627 change.
So obviously this is getting a lot of press, right? And so why is this? Should we worry about it? Or maybe a better question is,
I can tell you why it's getting a lot of press, but I bet. Yeah.
I want Richard to, are we ready for it spreading in humans? Let's put it that way. Do we have some preparedness?
There is some preparedness. I would venture that we're probably more prepared for an H5 pandemic than probably anything else. Are we prepared enough? Clearly not. But do we have antivirals that we know work against this virus? Yes. Do we know how to make a vaccine against this? Yes. Have we done clinical trials with H5 vaccines? We know sort of what dose we might need with or without adjuvants. Yes. So from that perspective, we're pretty well off. But again, with of course the conventional flu technologies, vaccine technologies, we're still not going to have a whole lot of vaccine apart from what's in stockpiles now for a number of weeks.
Well, and there's also the issue of flu vaccine efficacy.
That's right.
Which is not the 99% that we saw with the COVID-19 vaccines.
Yeah, but I think I'm going to put my flag out for flu vaccines a little here.
We're big fans here.
I think it's a little, the comparison's a little different, right? So the flu vaccine, you're comparing the efficacy of a vaccine in a patient population that has a lot of immunity to flu to start with, where at the beginning days of the COVID, it was nothing versus whatever you got from the vaccine. So it is a little bit different. I think should this virus take off, in my mind at least, the expected efficacy of an H5 vaccine, at least from very, very severe disease, would be higher than sort of the vaccine effectiveness estimates for seasonal flu. For that reason, it's a vaccine versus nothing type comparison.
So the neuraminidase inhibitors and the endonuclease inhibitors both work for H5 viruses, correct?
That's correct. I think there's maybe one sequence out there from one of these cow viruses that has a mutation that's sort of known to slightly lower the concentration, to slightly lower the susceptibility of these viruses. But in general, they're all very susceptible.
And then if we were to make, so the stockpile that we have, the H5 and the strategic stockpile, that's conventional inactivated flu vaccine, right?
That's correct.
Have people tried mRNA vaccines, and do they work?
So against H5, we've actually done a little bit of work with Scott Hensley and Drew Wiseman on this, not with this virus, but with a closely related virus where, at least in the ferret model, an mRNA vaccine was able to protect them very nicely. From the human perspective, the Moderna and probably others, but at least Moderna have presented some of their data from testing a seasonal mRNA vaccine in humans. The results were, well, it wasn't a whole lot worse than the current technologies, but it wasn't actually as sort of encouraging as I'd hoped, at least.
Mm.
So
These
Now,
are injected.
given that nobody expected this to show up in cows, is there anybody, or should there be somebody out testing other ungulates, wild ungulates like deer, to see if it is spreading in other places we didn't anticipate?
Yeah, no, I think that's absolutely. It's, we've seen this, as we talked about earlier, lots of different hosts, but we've seen some hosts we didn't know at all, for like dolphins and porpoises, cows. So, is it likely to be something else infected with this that we don't know? And right now I'm not batting at 1,000 on this virus and guessing where it's going to go. So I would say there probably is something else.
So I guess what types of things do you think we sort of should be looking for in thinking about its potential for a pandemic in the future? What things do you kind of think we should look at?
For this particular sort of where we're going with this virus?
Yes.
Yeah, well, I think, I think there's a lot to learn about how this virus is actually moving, about on the farms, because again, I think if this is truly not a respiratory spread, so it's doing something mechanical, at least in my naive mind, to me, if we can understand exactly how that's happening, then we can probably do something about it to stop it, right? It's maybe easier to implement some sort of advanced cleaning, disinfectant type thing than stopping a respiratory virus. So to me, that's pretty key in terms of reducing the threat of this particular virus. Again, I think following the trajectory of this virus in cows, of course, is also pretty key from a genetic level. We haven't really followed flu virus in cows before. So is it actually going to drive it to be more sort of human-like? Right now it doesn't look like it is, but I think it's potentially still early days. And if we start to see a sort of a shift of this virus from being more of perhaps an udder milk virus to then maybe being a little bit more respiratory symptoms, then that would be something concerning as well. So I think there's lots of really basic science to be done on this outbreak that can really, really help.
I guess that made me think, do the cows have viremia?
Probably, I don't think so. Could you detect virus in the blood of these cows? I wouldn't be surprised if you could, but again, looking at some of the data from sampling different parts of cows, it really does seem to be the milk. So yes, there could be spillover into that compartment, but I don't think it's a major part. Personally, don't think it's a major part of this disease.
Has this been transmitted to calves? Do you know?
So there's some reports of that, yes, where there are sort of cows on these farms that are infected, but it's not the majority. It seems to be the actively lactating cows, which is the dominant form of animal infected on these farms. So again, you say, well, don't the calves feed on those cows, of course, but which is true, but maybe that occurs before those cows really get into the milking parlour on a regular basis. So there's that period of time where they're not infected early on when they calf, and so the calf feeds, the calf's taken away, weaned, then they go into the milking parlour, that's when they get infected. So again, maybe why it's sort of circulating just within the
I was wondering if the
cows.
Perhaps different route of entry in a calf who's drinking from its mother's milk might have changed the pathogenesis of the disease, but that hasn't been seen so far, at least. Not on a great scale. Of course, some of the problem with this outbreak is we don't have as much information as we'd like from some of the affected farms. It can be challenging to get information from a lot of
personal and financial reasons of the people who own these farms, which you can kind of understand. But there are going to be, I think, some studies initiated now and more to come in terms of actual experimental infection of cows with this virus. So I think that is going to answer a lot of questions that we need as well.
And I think we'd probably also like to get, unless anybody has these data, some sense of how long the infection persists in the udder.
Yeah, that's right. That's key. Is key to both to sort of figure out when cows can move safely as well, right? So, how long does a quarantine have to be? When can you declare sort of an outbreak over?
Right. And I mean, from the dairy farmer's perspective, a dairy cow can be, 20 years milk source. And it's a major capital investment, and they're certainly not going to want to lose that.
No, absolutely. That's it's some of the problem, right? This is now, it's not a highly lethal disease in these environments. Again, from not at least what I've been told, this is not first-hand information for me, but certainly if a farmer's put on quarantine, they can't sort of milk or move cows. It's there's a lot of dollars per head that they're losing because of that. And up until today, when I think the USDA did come out with some thoughts of compensation for those farmers, there was none. So that really was an impetus not to report or not to want anyone to come and look.
Right.
May also be an impetus to start testing some of those antivirals in those cows.
Yes, maybe, and some of them might be a bit expensive, but there is precedent for sort of amantadine, the old versions of flu vaccines, and these viruses are still susceptible to those, being used in agricultural animals before.
Ask your doctor about Tamiflu. Yeah.
There have been something like 900 human infections with H5 viruses, right? And those are respiratory infections, right? So is that a different virus mostly from this one?
Yeah, that's a great question, right? So, yes, it's a different virus for sure. But does this virus have the capacity to cause severe disease? In experimental models, so, unfortunately, the ferret is fortunate for the ferrets. That is the sort of model of choice for us flu virologists. This virus is really, really nasty in ferrets when it's given experimentally. So it has that capacity to cause disease. If you look at there was a human case recently in Chile and also Ecuador. So with not this exact virus again, but very, very close cousins of this 2344B, they were more severe infections. So this is one thing I don't really understand about this. It's a really, really nasty virus in all of our sort of lab assays, but it's so much of the virus around, and there's relatively few human cases. So, it's things that don't really make 100% of sense to me. One doesn't seem to mesh with the other. I'm not smart enough to figure out what that is.
Oh, I asked Ron Fouché last year about his level of worry. He said he was worried about the wild animals, not humans, because he thought, like after the sea lion outbreak in South America, he's worried about wild animal populations being harmed by this virus.
Yeah, it's been truly catastrophic for some of them. I'm sure we've all seen some of those pictures of the sea lions and stuff on the beaches of Chile and Peru. It's wiped out many of them, and again, some of the bird populations as well. It's decimated some of them.
I think it's really interesting that you just said in ferrets, this virus it's the same virus from cows, right? That they've tested. It's lethal. It's a respiratory disease in ferrets, and it's lethal.
Yep, and
amazing.
No, I understand. Yeah, and that's very typical of this group of viruses. They all. So I don't. One other question I've got to ask is there something specific about this virus versus the other ones that are out there? I personally don't think so. I think this particular virus got in the right place at the right time, found its way somehow into the udder of a cow, and was all over. But I think that's also potentially. We'll get this from some of the experimental work as well, but I think that's important to know as well because if it's any virus can do that, that's a little bit of a different story versus it's only this specific virus or this specific trait that can get in.
Yeah.
Either way, it's a rare event. This is not. I'm pretty sure this is not something that's happened every year or every 5 years. Otherwise, the Europeans or the people in Asia would have picked it up before.
It's just I'm just interested because ferrets are the gold standard model for influenza, right? And here it's not for the one human case, which admittedly is just one, but it's not doesn't seem to be doing anything in humans. And I would say if there's so much virus in milk, it's certainly infecting some workers on cow farms. Yet the difference between humans and ferrets is very striking, right?
Yeah, absolutely. But at the same point, when we're infecting a ferret, typically we're dripping virus into the nose at relatively high concentrations, right?
Yeah.
So I don't know. Me, but it doesn't make perfect sense to me. And again, all these are models, right? So they do ferrets model probably a good percentage of some of what we see in humans, but maybe clearly not all of it.
So we should definitely make sure to not have large amounts of unpasteurized milk in our nose.
Yeah, that's not a good idea right now at all. Yeah, and so now we know we can take some of the milk from an infected cow and again administer it to mice, for example, and they succumb very quickly. So no, drinking unpasteurized milk is not the cleverest thing to do right now.
So
I don't think it's a clever thing to do anytime.
Well,
no,
right.
No, that's
we
right.
We had a letter a little while ago from somebody who's a big fan of raw milk, and we were a bit down on that for all the other risks, not specifically for the flu virus. Although it occurred to me after that, I mean, both my wife and my daughter eat raw oysters, and that's probably an even worse idea. But
Yeah. Well, I guess that's true. If people like raw milk as much as I like raw oysters, I can understand their hesitancy not to stop it. But in this case, it's not. It's really, 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 really risky.
Yeah.
So you must talk to the press quite a bit, right? Do you try and calm things down and not get them excited, or?
Yeah, it's a hard message, and I'm not very good at it, which is why I probably don't have a podcast or anything. But it's a real delicate balance because I think the risk really is low with this virus, right? It is a bird virus that it's not very infectious for humans, but on the the flip side of that, it's a virus I personally do not want to catch. And if of all the flu viruses that I've been here for 25 years at St. Jude and looked at a bunch of flu viruses, if there's one that I don't want to see in humans, this particular version of H5 is it. And so that the risk is low, but if that low risk turns into something higher and takes off, it could be catastrophic. So getting that balance across is really, really hard.
Yeah, it requires a nuanced understanding that not everybody has, right?
That's right.
But what you just said is you think the risk is very, very low. I think that's the important point, right? But also we are more prepared than we were for COVID, right?
Yeah,
we would hope so.
We at least know what it is.
Yeah. Are there other avian influenza viruses that you worry about?
So there's H5 aside, the sort of the H5N1 virus is one that always floats to the top of people's lists. For the reason it used to be probably the most widespread avian flu virus in the domestic poultry species. So it was widespread, again, apart from the Americas, but through lots of parts of Africa and the Middle East and Asia, lots of continuing human infections with H5N1. There's every year in China they detect a handful of cases of that particular virus, and it's one that is a little harder to see in action in the birds because it's not one of these highly pathogenic forms. So it doesn't automatically kill the bird. So you got to go looking for it. Much harder to actually find it. So that one is it.
When you said cases in China, you mean human cases?
Sorry, human cases. Lots of chicken cases, yes, but human cases, there's always anywhere from over the last few years, 2 to 12 and that type of range of detections of humans infected with that. It does tend to be young kids and often it can be people with underlying conditions, but all the same, it is a human infection. It's a chance for that virus to evolve.
So we did your paper last week on the H5N1 from bats, right?
Oh,
just last
yeah, that's right.
coincidentally last week, where we were trying to assess the the pandemic potential of that. So that went from a bird to a bat, right?
Yeah,
The
that's a really cool story. Sort of,
We thought so too.
I was going to
Yes.
say it sounds a little bit self-promoting, but yeah, we were lucky enough to be involved with some of the folks who actually collected that in Egypt, and we know bats have flu viruses, and maybe I'm rehashing your last week's episode, but
No, it's okay.
but those typical really bat adapted flu viruses are quite different to everything else. This one really did look like it was a sort of a chicken back to bat infection, and the fact that when it went to those bats and really picked up sort of this mix of avian virus-like traits and mammalian virus-like traits was looks really interesting, and does suggest we get sick of hearing about bats, right, and how important they are, but
Nope.
We
I don't
never get
that.
sick of hearing about bats
good.
and talking about bats here
Yeah.
on TWiV here.
So good. So yeah, clearly another reason that's a pretty important host.
I think, aren't there other avian viruses that have intermittently infected people, mainly at poultry markets, right?
Yeah, so there's a range, sort of the alphabet soup of flu viruses that have done this, from more recently H3s, some H10s, and again, you're dead right. Most of those tend to be in the environments of some of these live poultry markets where it's sort of a kid in a candy store. If you're a flu virus, you bring all these different hosts of flu together. They bring in their own viruses. They do their thing, and in those environments where there's a lot of chance for aerosolization as well. So yeah, there's a number of these, and you could sort of head to the WHO and look up this list, and that the list is growing, and I think it's a consequence of the COVID pandemic that there is more infrastructure globally to detect some of these sort of novel viruses now that we are picking up more and more of them.
So it's my impression that the global influenza surveillance apparatus is very big. It's much bigger than the coronavirus apparatus in bats, right?
Yeah, it is. At least on the human side, of course, the WHO has a global influenza surveillance and response system, which is, it's a well-tuned machine over 70 years, but that sort of focuses on humans. But there's also we're actually a collaborating center, a WHO collaborating center within that, where our focus is more on the animal side. And then there's actually at the sort of international agency level some good interactions with also WOA and FAO, who have more of the surveillance programs going in the chicken population. So yes, I mean, can we do more? Absolutely. Is there holes? Yes, absolutely. But is it better than most other viruses? Yes, absolutely.
All right, Richard Webby, St. Jude's Children's Research Hospital. Thank you for joining us. Really appreciate your thoughts.
Oh, you're very welcome. Thanks for having me.
This was awesome. Thank you.
This
I
was
learned so
great.
much.
Thank you.
Okay.
Brianne, I have to comment on this. I've been noticing it the whole podcast. The way you have your hair parted today, and with those earphones, you look like Princess Leia.
Yeah.
I don't know if that was deliberate, but it's
It
great.
wasn't deliberate, but I will take it.
Yeah.
Yeah, Princess Leia was pretty cool.
Yeah, I'm good with the
okay, yeah.
Princess
Now
Leia.
that you put them back up, and they're more obviously headphones, it's less like that.
Okay,
I think
well, it was like
partly...
they were slipping a little
Yeah,
bit.
it was because they went to the back, and those looked like, and they match your hair, and so it's like, wow. Okay, anyway, back to TWiV.
Let's take a couple of emails here. Hey, this one you should take, Alan, the first one.
Oh, from Peter.
Yes.
Yes, I should take this one. Peter writes, thanks for all your hard work, Vincent. Great podcasts. Attached is the correct way to pronounce apoptosis, according to the first pub. See footnote on page 241. Please tell Alan. Here's Peter. And Peter, thank you. As you will note from the way I just read your letter, I have taken note of this. Peter, by the way, is a professor in the biology department at Winona State University. So this is the footnote. This is from the first publication that used the word in a biological context. We are most grateful to Professor James Cormack of the Department of Greek, University of Aberdeen, for suggesting this term. The word apoptosis, and gives the Greek spelling, is used in Greek to describe the dropping off or falling off of petals from flowers or leaves from trees. To show the derivation clearly, we propose that the stress should be on the penultimate syllable, the second half of the word being pronounced like ptosis, with the P silent, which comes from the same root, to fall, and is already used to describe drooping of the upper eyelid. So it's apoptosis.
I once was told that one of the roots was the same as a root for pterodactyl.
Okay.
And I don't know the exact derivation, but I've heard that it was the same PT as in pterodactyl.
Okay. And here we go with the official original publication. So thank you, Peter. I stand corrected. It is apoptosis.
All right, we will say apoptosis from now on.
No more popping for me.
Okay. Brienne, can you take the next one?
Sure. Catherine writes, Hi, TWiV. Thank you for hosting such an informative podcast. I was introduced by the BI 115 class at Caltech. It was interesting to hear about the recent measles spike in your podcast. You mentioned people are getting conjunctivitis from cows that have measles, but it is not at the stage of human-to-human transmission yet. Why is that the case? Why isn't there human-to-human transmission, even though there are multiple symptomatic instances of cow-to-human transmission already? Given the many local outbreaks we've seen so far, I am also wondering at what point the government would step in and stop the spread of measles. How bad could it get before the government realistically intervened, and what would that government response look like? So I wonder if this is something similar to what we were just hearing about.
This is some confusion, I think.
Yeah, it's confusing.
Okay.
I think she means H5N1,
Okay.
not measles, right?
Right. I'm not really exactly
Because,
sure.
the cow, the conjunctivitis, human, that's just too coincidental.
That's
Right.
flu, and we don't have measles jumping from cows. Measles
No.
is a human virus.
Yeah. So
I think we talked about them on the same episode.
okay. Well, I think
Probably, yeah.
we just heard the most we could know about
Yes.
You heard from a
H5N1.
world expert.
Yes. So I think that is probably why we don't seem to have that level of transmission. And it seems like perhaps only one human has actually received H5N1 in this way.
Yeah. As far as we know, I guess you said, right?
Yeah.
Right.
Yeah.
And so the local outbreaks of measles are an entirely separate issue with an entirely separate virus.
Yep.
That is exclusively spreading in humans, and it is spreading in unvaccinated humans. And how bad would it get before the government realistically intervenes? Well, the government recommends that everybody get their measles vaccine, and everybody should.
Yeah, it's hard to imagine, given some of the pushback we've seen when thinking about some other viruses, what a more fervent government response might look like.
Well, right. Given that we do still have upwards of 90% coverage with measles vaccine, I don't think we need any more of a government response besides telling people not to be idiots and, get your kids vaccinated for measles, or they're going to get measles, which is a bad thing.
Recently Paul Offit on Beyond the Noise talked about the Philadelphia measles outbreak. I forgot the year, 90s maybe. And that mayor imposed a compulsory vaccination. Compulsory. It had never been done before, right? It's not just mandatory, you need it to go to school. You come here, we're going to give it to you, whether you want it or not. And that, he doesn't know if that stopped the outbreak because they moved into the spring, and that's when measles is no longer seasonal. But that'd be a tough thing to do nowadays, I think.
Yes.
People would. But the interesting thing is that there were two churches who were against vaccination, and when the compulsory order came out, the churches went to the ACLU to help them, and the ACLU said, you have a right.
Of luck.
You're out of luck. You have a right to refuse vaccination, but you don't have a right to martyr your child. So they said, nope, we're not defending you.
Yep.
That's very interesting, isn't it?
And the ACLU is hardcore about civil liberties. I mean, they have actually been on the same side as the NRA sometimes for stuff. They're not a liberal or a conservative organization, but they got to draw the line somewhere. And if it's your kid who can't consent to be unvaccinated, yeah.
And I think about things like that and realize that was before some of the immune amnesia information was understood.
For sure.
Yes.
Yeah. Okay,
That was
Sophia
also
writes.
before the internet was in widespread use and people could
True.
publish anything they wanted anywhere they wanted.
All right, next one is from Sophia. Dear TWiV team, thank you so much for providing such a valuable platform for those of us who are relatively new to virology. I'm currently taking a course at Caltech, Viruses and Applications to Biological Systems, taught by Professor Pamela Bjorkman, Professor Viviana Gradinaru, and Professor David Van Valen, BI 115. Listening to your episodes has greatly helped my understanding of virology and has been helpful in bridging the topics we learned in class with real-world virological studies. I had a few thoughts, questions from recent episodes that I would love to share with you, and if possible, hear your insights. In TWiV 1109, we discussed a paper on the loss of ORF8 in SARS-CoV-2. I was wondering, how could such deletions result in a selective advantage when they might be expected to weaken the virus's ability to evade host defenses? Could there be compensatory mechanisms within other viral components that improve fitness despite the loss of ORF8? Let's take them one at a time. So the authors say there must be a fitness advantage to losing it, right?
Right.
And in fact, they tested it to see if it was positively selected, and it looked like it was positively selected, which would be consistent with fitness gained from losing it. And then you say, well, isn't it an immune antagonist? Good question. Maybe there are others that do a good job, right?
Yeah, or maybe some of that immune signaling is also helping. You know, there may be both pro and antiviral effects going on with some of that immune signaling.
Right. Yeah. So the short answer, I guess, would be we know that that it seems to be positively selected. We don't know exactly why, but yes, there's some must be some compensating advantage.
I mean, this is the beauty of science, that you start to get little information, you get clues, you keep going, and maybe someday you figure it out. But that's the beauty of it, right?
Yep. Mm-hmm.
Next question. The H5N1 virus, which is mentioned, exhibits mammalian-like traits such as pH stability and ferret transmission, despite originating from an avian influenza virus reservoir. What mechanisms or evolutionary pathways might have allowed this virus to develop such mammalian-adapted traits while replicating in the bat host reservoir? Well, bats are mammals, right?
Yes.
So it, as Richard just said, it went from a bird to a bat and maybe bat to bat. And during that bat to bat, it acquired mammalian adapted traits. And that's why it's a bit of a concern, right? Because you don't need it to go into pigs or cows or humans. You got lots of bats around to do that. So that's why it's very interesting. Is that a good answer?
I think so.
I think that covers it. Yeah.
All right. In TWiV 1111, that's the last episode before the clinical update, we discussed the paper on using CRISPR-Cas9 to identify virus receptors. This is a really impressive and well-designed study that takes full advantage of the modern CRISPR-Cas9 screening technology. By using genome-wide CRISPR libraries and knockout cell lines deficient for other known viral receptors, the authors were able to identify MYADM and integrant subunits as host factors required for paraechovirus infection. However, one disadvantage of CRISPR screens is the possibility of off-target mutations introduced by the guide RNAs, which could interfere with the interpretation of the phenotypes observed. The authors performed extensive validation using isogenic knockout cell lines and complementation studies, but off-target effects can sometimes be difficult to fully rule out. Do you think any additional steps could have been taken to more rigorously control for off-target mutations and validate that the infection defects seen in the MYADM knockout lines were specifically due to disrupting MYADM itself? That's a good question. So, if I remember, they identified MYADM in the first CRISPR screen, and then they knocked it out specifically using a single guide RNA, same phenotype. And I think they put back MYADM
Yes.
and showed that now the virus can bind. Is that my...
I think that was, yeah.
If they didn't, that's what they should have done, right?
Yes.
Yeah.
You should always introduce the gene, because if it's an off-target effect, then putting the gene in would not have corrected it, right?
Right.
Yeah, and
Good
she mentions
question.
the complementation study here.
Okay.
Right.
Good questions. You should get full credit for those questions.
Yes.
Any insights into these questions would be appreciated and will help deepen my understanding of virology. Thank you for your continued dedication to educating and engaging with the scientific community. And I should add, beyond.
Yes.
We want to engage beyond the scientific community, right? Okay, we have a couple more. Let's go back to Alan.
Sure. Gail writes, Vincent, if I understand recent test results correctly, there are strands of viral DNA in various parts of the body of those suffering from long COVID, even though the strands don't comprise the full virus and the virus can't be grown. In the meantime, the components of H5N1, which have been found in non-raw milk, have similar characteristics in that they're DNA strands which can't be grown, RNA strands in both cases, which can't be grown at least so far and are therefore considered not to be alive, active. Since the strands of sorry, I'm translating into some terms here, but I get your question. Since the strands of SARS-CoV-2 seem to be correlated somehow with long COVID, isn't it possible the strands of H5N1, which people can get from pasteurized milk, could also turn out to be correlated with a possibly still unknown disease in the exact same way? If not, why would the dead particles from H5N1 be considered safe while the dead particles from SARS-CoV-2 seem to be correlated with extremely serious and dire long-term effects for many people? Okay, so my take on this would be the viral RNA that's seen in long COVID. We actually don't know if that's causing long COVID. It's just detectable, and what's going on with long COVID is complex and heavily involved with the immune response to a virus that had previously infected somebody in a fulminant infection that went to a viremia. Even if they had mild disease initially, that virus has kind of been everywhere.
Yeah, and my take on this, and I could absolutely be wrong. I'm thinking about some recent data I heard at a conference, but I could totally be wrong. Is that if there's SARS-CoV-2 RNA in long COVID patients, perhaps part of the idea that we're seeing there is that those that is an indication that there is hidden virus somewhere, and that RNA is just kind of like our indicator that there's hidden virus somewhere that we have not yet detected. And so, while yes, that RNA is not active or whatever term we were using, it's basically a marker that elsewhere in the body there might be active virus.
Right.
Here, when we're thinking about cow's milk, we're thinking about, milk is acellular. There are no cells. And so there can't be hidden active virus alongside of that viral nucleic acid. It's just viral nucleic acid that came from the cells in the cow. So the difference is with SARS-CoV-2, the nucleic acid is sort of a hint that there might be active virus elsewhere, but there isn't active virus in the milk. There's no cells for there to be active virus.
Right.
Does that make sense?
And yes, and the SARS-CoV-2, I would just add on to that, the SARS-CoV-2 in a long COVID patient is in somebody who had an active infection. The H5N1 RNA in the milk is something that you're drinking. And we drink vast quantities of all kinds of viral RNA with every meal, and our digestive tract just breaks it down as a carbon source. I mean, hey, yum, nucleic acids. So we're eating these all the time, and that's not something that's going to have any systemic effect on the body.
Yeah, I think one of the major ideas is that the long COVID is an inflammatory response, or the sequelae of an inflammatory response. And these pieces of viral RNA are really the remnants, as Alan said, of the original infection. I don't think they have anything to do with long COVID. Whereas the virus in the milk of cows is a shedding from the mammary gland, right? It's direct, and it's during the acute phase of infection when the virus is actively replicating there. So.
And if there is, as Brianne suggested, some compartment in which SARS-CoV-2 is actively replicating in long COVID patients, it's a compartment somewhere that we just haven't sampled yet.
Right.
Yep. All right, one more from Brianne.
All right. Tom writes, Dear TWiV team, over the last few COVID years, I have observed people talking about boosting their immune system by taking supplements like zinc and vitamin D. Even some legitimate healthcare websites, Fred Hutchinson Medical Center, Mount Sinai, for example, have statements about how zinc may boost the immune system. If I have correctly learned anything from Microbe TV, it is the human body and the immune system require a complex balance of many biochemical substances, many of which upregulate or downregulate others. But shouldn't that up or down regulation be a temporary response to an imbalance? It seems to me that the ideal immune system is one that is properly balanced. I understand that deficiencies of vitamins and minerals may make the immune response weaker, but shouldn't all we want to be proper balance, i.e. normal, neither deficient nor boosted? If I understand correctly, the COVID-19 cytokine storm is an example of an overly responsive immune system. Who would want that? Am I missing something? Tom is from Pasadena, California, and says, P.S. When you read my last email on a podcast, you said I was from Pasadena, but not adding California. FYI, there are also Pasadenas in Texas and Maryland.
I'm well aware of the one in Maryland. I actually had a grandmother, a little old lady who lived in Pasadena, Maryland.
Oh, I didn't know that there were others. That's why I just dropped it.
Tom, I think that your point there is a great point. I agree. And as an immunologist, I talk a lot about Goldilocks. You don't want too much. You don't want too little. You want just right. And I think that you've described that really well. I think that there are some pieces of evidence about certain nutrients at certain very specific times during certain microbial infections that might play a role, but I'm not sure that long-term anything other than balance and being properly getting proper nutrition is there's really anything other than proper nutrition.
Okay, that's it. We will do some picks now. Brianne, what do you have for us?
So when I was at a conference recently, there was a company called BIOS who is at the vendor fair, and they have a children's book. You actually can't get the children's book from their website anymore. I gave their website, so I have an Amazon link to the children's book, but it's called The Little Researcher. It is written for the 2 to 6 year old group, and it talks about a little dragon named Eliza. And the dragon named Eliza learns about the scientific method in this children's book. And it's very cute. I have since made sure to get copies for all of you 2 to 6 year olds in my life. And I think it's adorable and a great way to get little kids thinking about the scientific method in science.
Awesome.
Cool. That's great. Love it.
And proceeds go to the Discovery Museum in Acton, Massachusetts, which I have not been to, but it sounds like a good cause.
Yes, exactly. All of the proceeds do go to that. I forgot to mention that part.
Alan, what do you have for us?
I have a pick. It's not as cheerful a pick as Brianne's. I'm sorry, but I wanted to pick this, Vincent, while it's still current. And so this is a report. The Global Polio Eradication Initiative, which is a coalition of organizations, including the WHO, the CDC, the Gates Foundation, the Rotary International, all the folks who are trying to eradicate polio, whose goal I applaud, but whose methods I have often criticized. They commissioned a report to analyze how things have been going with this switch that they made in 2016, where they went to a bivalent oral polio vaccine, OPV. So this is based on the apparent elimination of wild circulating type 2 serotype of poliovirus. So they switched to an oral vaccine that has only types 1 and 3. They also have monovalent type 2 vaccine for when outbreaks may occur with type 2 derived from the vaccine. And Vincent and I have talked about this on past episodes. This, by the way, is not the point on which Vincent and I disagree, which is more about whether they should continue using OPV at all. This is a switch that they made a few years ago, and it has not worked out well at all. The report actually does a very blunt job of assessing that, and they say that this switch has been, quote, an unqualified failure. And so I'm linking to a news article in Science about this report, and it has comments from pretty much everybody involved. It's a report by Leslie Roberts, who's been covering this issue for a number of years for Science. And just you can read this article. It's accessible online, and you can look at the data and catch up on this.
So let me just explain. There's a little bit of a incorrect or omission here.
Yeah.
So as Alan said, we withdrew the type 2 component of OPV in 2016 because we hadn't seen any wild type 2 cases at all. And if you, I heard this from from Kara Burns from CDC at a meeting. She presented these data. If you look at the isolates of OPV type 2 after that, they go down for several years. But unbeknownst to everyone, a wild type 2 was still circulating in parts of Nigeria because there are parts that they couldn't survey because of terrorist activity. And so there was an outbreak of polio there. And so what the WHO did, they went in with monovalent OPV2, or maybe trivalent, I don't remember. But basically they reintroduced OPV2 into the population. And that's why it shot up, because now there's no type 2 immunity, right? You've stopped immunizing kids. So now that's why we had this huge increase. And to this day, we have lots of circulating OPV2 cases. So what do? That's why they decided to re-engineer OPV2 and make an OPV2. They said, this is going to solve the problem because it won't revert. Wrong. It does revert. It still paralyzes kids. It circulates. So not only was this decision in 2016 wrong, and by the way, Kara Burns said at her talk, we modeled it, and the modeling said it would work. But if you don't put the right data in the model, you get the wrong answer. Yeah. So not only was this wrong, but NOPV2 failed as well. It's a failure. Yeah. And so that's why I say we should use IPV
and my position is they should stick with trivalent OPV if they're going to continue using OPV.
Yeah.
I mean, I get, and I totally get Vincent's point that they should just abandon OPV entirely and insist on IPV. I take the stance that OPV is maybe a tolerable intermediate step if you do trivalent.
And maybe we could develop a better vaccine that has the mucosal immunization qualities of OPV, but not causing polio,
reverting.
Right?
Yeah.
But to do that, we'd need to do polio research, which we can't do anymore.
Which has been shut down because then you could potentially,
So the whole idea is that we can't work on polio because it's only in labs, but it's not. It's not only in labs anymore. It's out there. So it's
been out there the whole time.
It's been out there the whole time. We just didn't look for it. So why do we have to stop? Oh my gosh. That's cool. Thank you. That was going to be a
news item. Yeah, but I knew this was a guest episode and we don't do news items on a guest episode, but I wanted to get this out there. So that
I saw that. Yes, very good. All right. My pick is a channel on Instagram, an account called Errant Science. It's very wonderful graphics. I see them every day and I chuckle.
These are great.
And this one is types of PowerPoint slides. I love this. We have student, which is to be determined. It's a graph, to be determined. Then we have researcher, which is too many graphs. Professor, even more graphs in text. And then marketing is just the word future. Admin is a org chart. And the lab tech says, where's the overhead projector? But those are perfect summaries of PowerPoints made by different kinds of people. I love it.
The rest of this account looks great, too.
Oh, they're very funny. They're
overly
just
honest presentation questions and answers and bar charts really explained. Yeah, these are really fun.
And we have a listener pick from how do you pronounce it? Morag. Morag? Morag? I don't know. M-O-R-A-G-H.
Oh,
the G is silent. Mura. Hi, dear TWiV team. If nobody has already suggested this for a listener pick, may I please recommend Paper Cuts podcast with Miranda Sawyer, Alex von Tunzelman, John Elledge, etc. We read the papers so you don't have to. It's 20 to 30 minutes of a small, intensely funny rotating cast of UK comedians, authors, journalists. They read some of the day's headlines in UK papers, riff on the stories. The humor can be raunchy but never cruel. There are F-bombs dropped, but frankly, news with swearing is the only way some of us can bear it. The topics range from frivolous, royal family, food trends, celebrity gossip, to profoundly moving, a special episode on UK newspaper coverage of the October 7th Hamas attack, and everything in between: the Royal Post Office scandal, the recent measles outbreak in UK, a whole lot of Rishi Sunak and other UK politicians' frequent soccer metaphors. Paper Cuts is the only other podcast I listen to so far that matches TWiV's effect on me. Routinely cracks me up laughing, teaches me things I didn't know, and feels like, as Will Newcomb just commented, a family gathering. One of their co-hosts has been comedian virologist Rialina, a PhD in viral bioinformatics from University College London, but she has already been a listener pick, TWiV epitope 852, Tree Man Syndrome, from listener Chris. But I'd like to recommend her again, along with the Paper Cuts show, for her tireless and persistent willingness to be the guest boffin on TV, radio, podcasts, etc., who clearly explains immunology and how vaccines work and why they're important, while also being incredibly funny and honest. I hope she might even be a guest on TWiV someday.
Sounds like a great idea.
Sounds like a great idea. I'd love to have a funny person on.
Well, none of us are funny.
Many thanks for all you do. Love the banter and the digressions. Mora Mo Ramaj, my alter ego is an eager alto. Thank you, Mora. Morag. That's
Mo.
TWiV.
Mo.
Thank you, Mo. That's even better. Yes. That's TWiV 1113. You can find the show notes at microbe.tv/TWiV. Questions, comments, pics, TWiV at microbe.tv. And if you enjoy our work, consider supporting us. Go to microbe.tv/contribute. Oh, I was about to say Dixon de Pommier because he's here all the time, but he's not here. How about that? Alan Dove is at alandove.com and turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
Brianne Barker's at Drew University, BioProf Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.